We were studying the process of transcription. In this class, we will take up the different steps which will be seen in the process of transcription. We have already got the gist of transcription, how the DNA will be transcribed into RNA. Let us see it through the steps. The transcription process takes place through 3 steps. There are 3 steps involved in the process of transcription. The first step is the recognition or the initiation of RNA synthesis. Recognition, from where the transcription will start, the place or the location or the region of the DNA from where the transcription will be starting, so that the way we will get a promoter. Because once we recognize the place where transcription starts, then we will have a promoter. Promoter will be attached there. Okay. So now once we recognize it, promoter will be attached and RNA will start synthesizing. The second step is elongation of nucleotide chain. The second step here is the elongation of nucleotide chain. Lengthening of the nucleotide chain will be taking place. Okay. And the third step is the termination. Transcription process takes place in 3 steps. Recognition or initiation of RNA synthesis. The next is elongation of nucleotide, and the third is termination. We have already established the unit. What is the unit of transcription? That is, unit of transcription consists of a promoter, structural gene and terminator. So let us see how this process will takes place. So here now, this for RNA synthesis, that is transcription process. That is forming of RNA from a DNA. We require the enzyme RNA polymerase. See here, many times we are discussing this one. So this enzyme is very important for RNA, that is transcription. Without RNA polymerase enzyme, transcription will not take place, because RNA will bind at the promoter site. RNA polymerase will bind at the promoter site. So here, during this, let us see the step by step processes. That is, the first step is recognition or initiation of RNA synthesis. For example, this is the double stranded DNA. Here coil it is there, I have removed this coil here. So this is the double stranded DNA. So the black one is the coding strand. Black one is the template strand. Okay, it is from 3' to 5'. The black one is from 3' to 5'. I'll write here 3' to 5'. And this blue one is the coding strand. Okay, so this is the coding strand and this is the template strand. Coding strand is from 5' to 3' direction. So here, this is the DNA. In this transcription, only a segment of DNA will transcribe into RNA. This whole DNA is not, only a part of the DNA. This whole DNA, a part of the DNA is transcribed into RNA. So here, first it will be recognized which region of DNA will be transcribing into RNA. So the region will be recognized. So here promoter will be attached. When the region is recognized, that from this place to this place, from this place the transcription starts. So at this place a promoter will be added. Once this place is recognized, promoter will be added. Okay. So here, once we get the promoter, this RNA, that is core enzyme, RNA core enzyme will be attached along with this core enzyme. Now, one more thing is there which is important, sigma unit. Okay, this core enzyme, along with this core enzyme, sigma unit will be added. So it forms the holozyme. When this core enzyme, that is RNA polymerase core enzyme, along with this sigma sigma particles will be present. Okay, these sigma particles are very very essential in the initiation of the initiation of transcription process. This sigma subunit, sigma is very important in the initiation of transcription process. So, the sigma unit along with this core enzyme, this together makes the holozyme. Holozyme is nothing but core enzyme, that is protein part plus cofactor. This cofactor is nothing but non-protein part. To this core enzyme, that is, enzymes are nothing but proteins. Enzymes are made up of proteins. To this core enzyme, sigma is added. Sigma unit is added. So, to this core enzyme, the sigma unit, which is cofactor, is added. Okay. So, this is the non-protein part. Non-protein part is very essential. Okay, along with this protein part, non-protein part is also essential. So, it makes the holozyme, which is nothing but core enzyme, that is protein part plus non-protein part. So, it will get attached at this promoter site, this RNA polymerase. This is the RNA polymerase, okay, plus sigma unit. This sigma unit is also very important in the initiation of transcription process. So, they will bind at the promoter region. So, next comes when it binds na. So, this is the first step where initiation. First, we will be recognizing from which region of DNA. Because out of this whole, it might be this region, or it might be this region of DNA, or it might be this region of DNA. Which region of DNA is going to transcribe? First, it has to be recognized. As soon as it is recognized, promoter is added, that is, initiation of RNA synthesis will take place. To this promoter, RNA polymerase enzyme and this sigma unit is added. So, this is the first step of RNA, that is transcription process. Now, after it is added, comes the second step, which is nothing but elongation of nucleotide chain. Elongation of nucleotide chain. In the previous class only, I have explained you people how nucleotide will be elongated. So, this one is there now, the RNA polymerase enzyme is there. So, here to this polymerase enzyme, nucleotide will be added. RNA nucleotide will be added. So, this polymerase facilitates opening of helix. In the second step, where elongation of nucleotide is taking place, what is happening? The polymerase facilitates opening of helix. As soon as the site is recognized, okay, from here transcription is taking place. So, what happens? Till now it was coiled, now DNA was coiled. As soon as we recognize the site, now here what happens? This helix will start to open up. The helix will start opening. Okay. So, this polymerase enzyme will be moving. After adding this nucleotide, it will be moving again. Then again nucleotide. So, it will slowly starts opening the helix and adding the nucleotides. It will be starting opening the helix along with this addition of nucleotides. Because now it has opened here, it will move here. Then add the nucleotide. Then again it will open it. Then again add the nucleotide. Like this it will go on traversing on the template strand. Who will go on traversing? This RNA polymerase will go on traversing. Opening of helix. Then it traverses. It moves towards the strand. Slowly moving. This RNA polymerase was here, then it was again come to this place, then again come to this place, then polymerase will move here. The enzyme will be moving, and when it moves, it will open the helix as well as add the nucleotides. So it traverses template strand. Traverses means travels on the template strand. The RNA polymerase, it was first here. Now it will move to this. It is traveling from here to here to here to here. It comes till here. So RNA polymerase facilitates opening of the strands by traversing on the template strand. So when it traverses, thereby creating a, because as it is opening, here addition of nucleotide is also taking place. When the strand is opening, nucleotides are added. Nucleotides are added. So when with the addition of this nucleotides, it creates a RNA copy. So it creates a copy of RNA. It creates a copy of RNA. So after creating the copy of RNA, it proofreads. After creating this copy of RNA, it proofreads whether the sequence which has been formed, whether this copy complementary strand, that is RNA strand, whether this complementary strand is having the correct nucleotide sequence or not. The sequence will be proofread. Okay? Once the sequence is correct, once it sees that the sequence is correct, what happens? Here, the next step will take place. The next step is the termination of transcription process. Okay, so here now for initiation process, along with this RNA polymerase enzyme, one sigma subunit was required for initiation process. Now for this termination process, we require Rho factor. We require Rho factor. Rho factor will be helping in the termination process. Rho factor will be helping in the termination process. Once it will move towards this 5' end, it will move towards the 5' end, what happens? There will be Rho factor. Here it associates with the Rho factor. Here now somewhere Rho factor will be present. So this polymerase enzyme combines itself, associates itself with the Rho factor. Okay. Now here for example, if Rho factor is present, we have this transcription, Rho factor will be there. So as soon as it sees the Rho factor, what happens? The transcription starts. As soon as this DNA gets this Rho factor, the transcription will be stopped. So now transcription has been stopped because this polymerase enzyme has encountered has associated with itself with the Rho factor. As soon as it has associated with the Rho factor, here the enzyme, the process of transcription has been stopped. Now the formation of RNA strand has been stopped. After which what happens? As soon as this Rho factor is associated with the polymerase enzyme, here this Rho factor destabilizes the interaction between template strand and RNA strand. This is the template strand and this is the RNA strand. So a connection is there. So new nucleotides are being formed. As soon as this RNA polymerase gets associated with the Rho factor, now the connection between these two strands will be removed. The connection between these two strands will be removed. So destabilizes interaction between template strand and the RNA strand. So now there is no connection between this one. The RNA, it has destabilized. Connection is been gone with the help of this Rho factor. Now this RNA is separate from this template. It is where it is inside now. Now RNA comes out. RNA comes out. So this is how this RNA strand has been formed. RNA comes out. So where this whole process is taking place? Inside the nucleus. So it was here, now from here it will come out inside the nucleus. The strand was here inside it, now it will come out from here, it will come out from the nucleus. Sorry, it will come out from here and it will fall on, it will be in somewhere in the nucleus. Afterwards it comes out from here, it will be in somewhere in the nucleus. and it won't be in the nucleus for long, because it will be coming out of the nucleus in order to give the message, because RNA has taken the message from the DNA, coding from the DNA. According to these codes, DNA, that is protein, will be synthesized. According to these codes, proteins will be synthesized. So here it will be coming out from this, it will be present in the nucleus, then from the nucleus also this RNA comes out and helps in the synthesis of proteins. And after the process is over, here the polymerase enzyme dissociates. After the process of transcription is over, when this has come out, when this RNA has come out, when it is in the nucleus, then RNA polymerase will dissociate, because the work is done. There is no more work of RNA polymerase enzyme. So once the transcription stops, the RNA that is polymerase will be dissociated. So this was how transcription process takes place. That is, from DNA, from one strand of DNA, a new strand of RNA has been formed. Okay. So from the DNA, a copy of RNA is formed. So this process in which from this template strand of DNA, a copy of RNA is formed. So this process is called as transcription, which takes place in 3 steps. That is recognition, elongation, that is initiation, elongation and termination. So this was all about the process of transcription. In the next class, we will start with another process, that is translation. I have explained you in very detail. I have repeated all the sequences, all the process, all the events which takes place many times. So we have finished with this process of transcription. Next, we'll start in the next video, we will start with the process of translation.